Good morning to everyone, and welcome to the Primetime at the BU Library. Primetime is designed to celebrate the experiences and accomplishments of Bethel faculty, students, and staff. It's a collaborative project between the Friends of the BU Library, faculty development, and other offices on campus. We record most of our programming, and you can find the recordings in the BU Digital Library. Join us on Thursday, December 8th, as Edgar Scholars Professors of Nursing Ann Holland and Joan Tiffany and Nursing Senior Ashley Coleman describe the results of a pilot study to test the effectiveness of training intervention on nursing faculty evaluating student performance in simulations. Today we welcome Dr. Teresa DeGolier as she shares her study of the effect of herbs on isolated uterine tissue in a presentation, Herbal Supplements and Uterine Contractions Still Crazy After All These Years. Okay, let's welcome Teresa.
All right, thanks for being here. There's kind of a cloud of sound here. My goal is to kind of catch you up with what I've been doing for research. Several of you have asked me what work we've been doing with students, and there's a lot of interest in the midwifery program that Bethel has. Is there use of herbs that's valid for telling people how to promote labor or augment labor or prepare the womb? So this will not be so much science as it is what have we learned about this process. So there are some results, but I'm not focusing on lots of the science behind the results. Just what have we learned of interest? I start out with a slide like this because clearly I think about one of my colleagues right now. Melissa Cordes is pregnant and is due. And is she on campus today?
Yes.
All right. So I was hoping if she's here, I was thinking, I hope she's not going through this. Things have certainly improved. But I did talk to her yesterday. She said, what do you think of red raspberry? I've been taking red raspberry tree. I've got to get this baby out of here. I'm kind of sick of being pregnant. So we talked about that, and hopefully she'll have a good outcome. But the whole point of what we're looking at is to try to see is there any validity when people use herbs to promote labor. So here is the next slide. Here is the next slide. There we go. And let me give you some extremes. Okay, historically, using plants for medicinal purposes goes way back to a lot of Native Americans. Eclectic physicians are a branch of American medicine, not so popular now, but basically what they said is something works, you use it. And so a lot of plants were there. Here is a current study that some of our students are doing on, which is calabash gourd, which is found in Belize. And from a book that some tropical scientists have put together, they interviewed Mayan medicine men. And then they compiled some data. They said, you know what? The Mayan medicine men tell women that if they want an abortion, you should eat the seeds of this. And if you want to, excuse me, induce labor or the afterbirth, eat the fruit. So we managed to get a hold of one of these gourds, and we've been testing it on isolated fragments of the uterus and coming up with some very interesting results. The fruit indeed does contract the uterus, but the seeds are not very potent. Now in contrast, here is a quote from a book that's very common for people who like to read about midwifery. And some midwives do employ different types of herbal cocktails. And this is kind of a formula for one. Here we go. 2 ounces of castor oil, 2 ounces of vodka, and 2 ounces of orange juice. Now, orange juice is going to help everything to taste good. That's all it's doing. Okay? The vodka is just for relaxing. Melissa, so glad you're here. Yeah. All right. Then the castor oil, you go, no, I don't want to have castor oil. That's going to give me the runs. And most people have thought for years that the irritability of having intestinal runs was enough to promote labor. But a lot of information is now available that says, you know what? We use castor oil. Pancreatic lipase breaks it down to a fatty acid called ricinoleic acid, which is a little more soluble than some fatty acids. So it goes in the bloodstream and actually binds to prostaglandin receptors on your uterus and causes contraction. I was sharing this with Melissa and she said, can't we just take the ricinoleic acid by itself and not have to chase the castor oil? So we have both extremes. We have medicine men and we have contemporary clinical situations where herbs have been used. If you look online and want to get some ideas of what are some of the more common herbs that people use to, again, promote labor, induce, augment, prepare the uterus, these are some of the more common ones that you would find. Surely you've probably heard of blue cohosh, black cohosh, castor oil, as I've mentioned, red raspberry, evening primrose oil. There are several. Here's some pictures. Here's juniper berry, which some of you may have in your yard. Have you ever thought about those berries as something that could induce labor? Or this right over here is shepherd's purse, just the whole plant crushed up. So quite a variety of things that are readily available for us. So the question I ask is why are folks doing this when we have such good Western medicine in the U.S.? Why are we doing this when we have hospital settings where Pitocin and prostaglandin products can get labor going? And for a lot of people, they feel that those are dangerous. There can be some side effects if things aren't going well, and many people want to take a more natural approach. And then people don't want to take a natural approach because there's not enough research saying that this makes any sense and is it all a placebo. So that is kind of where we are coming from on this project. Is there any validity to these claims? So in 2007, a student, Jennifer Berger, said to me, seriously, can we test any of these products like blue cohosh and to see if it actually induces labor? She's a doctor now, so she kind of had that mindset. And I, being a biologist, decided, well, let's play the role of the skeptical scientist. Go, I don't know, let's find out. We have the tools to do this. So we decided that we would just use herbals as they're available to a consumer, like go to a grocery store, or go to a drugstore, or GNC, and see if we can't find some of these herbals. Mix them in an aqueous solution. Make it real easy for them to be ingested. But we would test these herbals on isolated tissues. It would look something like this. This is called a smooth muscle bath. And right here, the red strip that you see is a piece of the uterus from a mouse. So we took it out of the animal. And then what's outlined in blue is the same thing as the animal's extracellular fluid. So we're creating this artificial environment. We bubble oxygen in there. We have a little sucrose in the fluid so we can keep generating energy for the tissue. We hang it under a tension that's consistent with how these organs lay in the body. We can control the temperature so that we're at the same temperature of the organs in the body. So we've got this very viable piece of tissue. And what we've done is we've taken it out of the body, though. So there's no influence from the central nervous system. So we took the head out of it, all right? No influence of the central nervous system, and no endocrine control. Just plain smooth muscle from the uterus. I said, well, we can do this. We can take mycin. Ready for this? Here we go. It's exactly what we did. All right? This is the abdominal pelvic cavity. The mouth's opened up. Mice have uterine horns. There they are, 1, 2. We just snipped them out, hung them up under tension, put them in this bath, and now we have just uterine tissue. And this is what we tested. We basically took our herbal materials of interest, mixed them in a consistent concentration base, and then basically just pipette them in. And then we wait to see what happens. This is what happens. This little movement you see right here is a natural, spontaneous rhythm that uterine tissue has. If you stretch it, it'll actually start to contract all by itself. So that's a good sign that our tissue is healthy. Then, in this picture, we've applied a botanical. We've applied blue cohosh. And we go, oh my goodness, look at that. This line going up measures an increase in force, which means the tissue is contracting. It not only goes up in force, you can see that the previous amplitudes have increased in both frequency and force. And so those are kind of the markers we're looking for. When we put these drugs on these tissues, do we get increase in force of contraction, in the frequency of contraction? And sometimes we get something called a change in basal tonus, which is when this baseline finds a new area. It's kind of like the tissue's going, and staying there. So that's what we've been looking for. Now, if I back up and follow my script here a minute, what I'm going to attempt to do for the next 20 minutes or so is say, after approximately 9 years of doing this with several different herbals, step back, what have we learned? Okay, what have we learned? All right, first of all, they're all potent. Every single one we have tested contracts the uterus, which really surprised me. I want to take the skeptical role and say, oh, this is not placebo effects. You know, if you tell someone it works, the power of the mind and all the neurotransmission that goes on, well, sometimes these things happen. But we took all that away, and my goodness. If we can trust that when you take this orally, it's actually absorbed and finds its way to the tissues, it will contract. So we got excited about this and just pushed through several botanicals. What else did we learn? How potent is potent? What does that mean? Well, how strong is strong? And how does this compare to some of the natural stimulators of the uterus that the body would have? One would be acetylcholine. You have neural tissue that releases a neurotransmitter called acetylcholine, which should contract the uterus. So if we do acetylcholine on the same tissue, flush it out, let the tissue return to its normal inherent rhythm, and this would be evening primrose. All right, look at that. It's not quite as much as acetylcholine, but close to it. So what we did is to try to standardize this by using consistent concentrations of our herbal mixes to consistent, in the case of acetylcholine, since it's a single molecule, molarity. So we have a standard to compare all our contractions to. How does acetylcholine compare to oxytocin? And this would be the same dose of acetylcholine here, different tissue, different experiment, different y-axis because it's on a different set of measuring tools. But nonetheless, same concentration of acetylcholine compared to oxytocin. Oxytocin is a little higher, and that dose would have been 10 to the negative fifth, which is quite a bit stronger than the physiological levels in your body. All right, quite a bit stronger. But nonetheless, we're trying to get some reference point to compare these two. So we see that, wow, this is oxytocin, the body's normal hormone to help promote labor, uterine contractions during labor. And nice, strong contraction, somewhat stained with an increase in frequency. That's what it looks like on a single uterine horn. So this is when we say how potent is potent. Well, what we would do is one of two things. We would take these heights and express them relative to acetylcholine, or these heights and express them relative to oxytocin. Trying to get a sense of how potent is potent. Okay, on the x-axis are increasing concentrations. Now notice they're not molarity because this is a cocktail of compounds. These are plants. Who knows what's in there? Maybe hundreds of chemicals. So we simply did concentrations. Milligrams of plant in a 15 ml muscle bath. And when we increase doses, would we see a concentration response? Sometimes yes, sometimes no. Here's a project from a few years ago by Nick Kwan, where we have a lovely concentration response. The higher the dose, the higher the response, up to a point where it plateaued. Very typical. Here is a student, Amy Olson, from a few years ago. Red raspberry. There's a little bit dose dependency, but overall, everybody's just kind of hanging around right here. And if you look at the y-axis on both of these, notice that both of these plateaus are really close to about 100 what? 100% of what acetylcholine does. So we found that usually at approximately 20 to 30 milligrams of concentration, we would consistently get a contraction that was about the same as our standard dose of acetylcholine. Which is what this slide. What did we learn? That at equal concentrations, particularly about 20 to 30 milligrams in our bath, everybody is very close to what acetylcholine does. This is 140% of acetylcholine. Of all the things we've tested, castor bean is the highest. Blue cohosh and red raspberry leaves are very high. Evening primrose not quite as high, but you know, when you look at those standard deviations, they're not different from each other. So these are fairly common herbs that have been used, sometimes in midwifery, sometimes just, I'm going to order these online and do this myself. They're fairly common, and people have had success with them. Just throwing some pictures in here for you. These are evening primrose seeds. So we didn't use oil. We can't use the actual oil because the muscle bath is aqueous. They have to mix. So we got seeds and crunched them up and then tried best we could to put them in solution and filter out the debris. One shortcoming of all this is we really don't know how much we have in there. All we know is we have an aqueous extract and we're trying to test based on that aqueous extract. Now, clearly they contract isolated uterine tissues. What if a consumer decides, well, I'm an intact person. I'm going to take some blue cohosh or castor oil or red raspberry tea. Should I be concerned about systemic effects? I've only done this now on one herb, but notice, are there systemic effects? Excuse me, yes there are, which means there are side effects. Oops, right away, this becomes a concern, because none of this is FDA approved. As long as nothing has shown problems, the FDA is not going to investigate. If something works and people have success, then stand back and go, more power to you. So here's what we have. What we did is we took blue cohosh, and this is the one herbal that we have spent the most of our time with, and we tested it on several other tissues in the body in a similar manner, isolated tissues. And this is what we found. Blue cohosh on the heart is going to stop it. It'll slow it down, it'll stop it, and we have found since then that some folks have done some research at the level of isolated, I shouldn't say isolated, embryonic developing rodents and have had cardiac effects. And there have been people, unfortunately the thinking here isn't always good, but if I take blue cohosh to get contractions going, if I take more, they'll go more, right? That's kind of their thinking a little bit. Well, the more you take, the more it's going to bind to other receptors, and some of these people that have taken too much blue cohosh have had babies born with cardiac birth defects. So there are some issues here. What does blue cohosh do in the aorta? Absolutely nothing. We thought we'll try vasculature. Didn't do a thing. But that doesn't surprise us, because the thinking is that maybe the biological constituents of blue cohosh bind to what are called cholinergic receptors. If they contract similar to acetylcholine, they must bind to a receptor similar to acetylcholine. Well, those receptors are not found on the aorta, so that made sense. Stomach, ileum, colon, urinary bladder, vas deferens were other tissues we checked out. Stomach, ileum, colon, all contracted. This sounds like blue cohosh is binding to something that promotes peristalsis. And for those of you that have had some of the biology background, it sounds like a parasympathetic response. Urinary bladder, whoa, that was pretty important. That contracted with quite a bit of force. But let's be honest, I presume if you're having a baby, you really don't care if your bladder is empty. All right, but it's a powerful contractor. Here, we thought, in all fairness, let's do the vas deferens, okay? Not good. Okay, it's just not a big contractor, less than 25% of acetylcholine. So the conclusion my student had was this, is you would not take blue cohosh to advance your sexual performance. I thought perhaps we should rephrase that on your paper just a little bit. All right? This becomes interesting because it helps us to begin to think about what is in blue cohosh that's actually doing the work. It's a plant. If we could only determine who the active ingredients are, then we could isolate the active ingredients and begin to figure out who's actually doing what and how. So these M and N simply stand for different types of receptors that are found on tissues. In this case, the constituent alkaloids, which I'm about to show you, have been shown to bind to cholinergic receptors in the central nervous system. Our results would suggest that something's binding to receptors in the peripheral nervous system. So we got Trey Maddox involved from chemistry. And Trey says, hey, I can do this for you. I can take actual blue cohosh, the roots and rhizomes is where these products are, and I can crunch them up, and I can actually extract out what are thought to be the chemical constituents that may contract the uterus. So he did that, and he gave them to us. So we tried isolated agents, and these are the results we get. These four are thought to be the big players. N-methylcytosine, I'm not putting values on there like millinewtons of force. I'm just saying plus like, yeah, it's a contractor, but it's not very strong. Very small contraction. Baptifolene, a little bit bigger. Anagyrine, a little bit bigger. We haven't done magnifolene yet because its chemical structure is a little more difficult to pull out. So we looked at these and thought, you know, they're players, but when we compare their contractile forces to blue cohosh himself, they don't add up. It's not that big. So another group of compounds that supposedly might contract the uterus are the saponins. And right now we're doing saponins, and they're powerful contractors. But saponins, and some of you may have used these in labs, are permeating objects. They'll go inside the cell membrane. Well, of course, you do that. Here comes calcium. Now we have contraction. So we're beginning to look and go, okay, we're starting to see who's doing the job here. Who's doing the job? We could do this with several other compounds. I'm not sure I'm really interested in pursuing that in great detail. But what we've learned from this is, boy, if we know who the individual compounds are, then we can do what are called receptor antagonism studies. We can try to block the receptor and see if the effect of the herbal no longer happens. But it's very frustrating that I do that with the plant itself. We can't do it. We really need the isolated agents. Okay, what else have we learned? Okay, we've actually learned that when we buy stuff from the store, it is what it says it is, which actually surprises me. And unfortunately, I came across this the hard way. I tried to publish something, and one of the editors said, this is all very interesting, but can you verify that what's in that container is what's in that container? You know, if I pick up a bottle of red raspberry crushed up leaves and nice little cellulose caplets, he says, how do you know that's just not a bunch of sand and calcium? Because that'll contract the uterus. I go, well, I don't know that. I'm just raised to be this honest Midwestern girl. I believe anything that comes on the label. I mean, that's kind of how we think, right? So, Paul Sonerel and a student that graduated last year, Elliot Blankesmore, said, we can help you with this problem. There's a molecular technique called DNA barcoding. And basically what they can do is they can take the substance and then do a bunch of molecular techniques and express the DNA. We can send this off to an institution or a research group, and they do something called BLAST, which means I read the DNA and I compare it to a library of known DNAs, and then it comes back and says, the DNA that you just sent me contains the following things. And wow, what we found out is everything we have tested was what it said it was, except one, black cohosh. And of all things, black cohosh was inside the chemical compound, but so was cilantro. What's that doing in there? Well, which says, do we have a quality control issue? Okay, and the black cohosh that we had was purchased online, presumed for the US, but we were able to trace that the cilantro came somewhere from Southeast Asia. Now, you know, when you're working in harvesting on this stuff, I can see how easy it is, especially if you're not a plant taxonomist, to get these goofed up. I'd be very curious if cilantro actually contracts the uterus. Haven't tested that. It would smell good at the very least. Okay, what else have we learned? When possible, get the whole plant. Because then I know what I have, right? If I can get the whole plant. We started out all these research projects by simply going over the counter to the drugstore, to GMC, or online and ordering them. Now, we feel kind of good now that we know that basically what we ordered is what we got. But since then, we tried to stay away from that and say, let's just get our own stuff. So we've repeated a few of these. Just go up north, grab a bunch of red raspberry leaves, crunkle them up, and repeat the experiment. Castor seeds. We've simply have ordered the seeds instead of a gel. All right, evening primrose. I can get those up north. Just stop along the highway on 35 when evening primrose is blooming and grab the seeds, and there we go. So now I can actually say I have the equivalent of an herbarium stock sample, all right, a voucher specimen. Calabash fruit we got from Belize. The human kinetics folks have a course that goes to Belize, and they set up with the field station there to send me a calabash gourd. Ginger root, and there's a lovely ginger root right here. All right, made the lab smell really good, but not a very good contractor of the uterus. Okay, not very strong. This is squaw vine. Again, I keep putting a few of these pictures up here so you can kind of recognize some of them, but we've learned we're much better off if we get the whole plant, because then we actually know what we have. What else have we learned? We're pretty confident that most of the herbals that we have used, in some manner, trigger what's called prostaglandin synthesis. All right, and this is kind of just a black and white sketch, but it basically says that a number of chemical ligands can bind to receptors, which further downstream will promote the formation of prostaglandin, which of course is the body's, another body chemical to contract the uterus. So you see on here acetylcholine receptors. A lot of the materials we've used, since they provide a contraction equal to acetylcholine as possible, they're binding to acetylcholine receptors. Catecholamine receptors, we feel that red raspberry may actually be working through that because we've been able to block a little bit of that. Other objects we use don't work this way. The saponins basically permeate the membranes. It's probably true that red raspberry may also just work because red raspberry leaves are really high in calcium. You want a calcium force? Throw some of those in your salad. Really high in calcium. Maybe it's just high extracellular calcium that's doing this. Just so many unknowns. The other thing we've learned is when we get all excited about telling people like, yeah, these things contract the uterus, this is awesome. You really got to stand back and go, so what? Because this is not how any one of us experience this. We have not donated our uterine strips and expected to put them back, right? This means absolutely nothing unless we know that when consumed from an organismal perspective, it's absorbed and actually finds its way to receptors. Now, based on what we've done, I have a tendency to think this is true. Somebody else's job, perhaps a biochemist or a molecular biologist, is going to be able to go, all right, let's take urine samples and see if we find metabolites of these plants. Then we have some confidence that the material absorbed in the urine represents what's been filtered out of the blood, so that would be good. The placebo effect remains a skeptical piece. For a lot of people will say, this is all in your head, you feel good about it, it's going to work, and we all know that placebo effect is helpful. Some of our moving forward when we don't feel good is because I want to try and I can do it, this is going to work. But if we could say with some confidence there really is some science behind this, this would be great. To give you an idea, I mean, I want to call myself a skeptical biologist because I do believe that science advances by rejecting the null hypothesis, right? But I look at some of this and go, we should be paying more attention to this. I'm going to read to you 3 very short paragraphs from this wise woman herbal thing. Do we have nurses here? Do you know this book? I got it from one of your midwife people and said, you got to read this. Because they were like, really? I'm like, really? Well, they come together. This is kind of weird, but just bear with me a minute. This is a labor tincture, okay? We need a ½ ounce of black cohosh root. We need a ½ ounce of blue cohosh root. We need a 1/4 ounce of ginger. We need a 1/4 ounce of birth root, which is trillium, all right? And then we need 1 cup, again, we need more vodka, all right? Now, here's the directions. You might want to make this for Christmas holidays. Maybe you don't. No, okay. Place dried herbs in a pint jar and add your vodka. Label, good. And cap. Let it all seep together for 6 weeks or longer. Decant the tincture by running the mixture through a juice extractor or a salad spinner. If neither of these is available, pour the tincture and herbs into a cotton cloth and wring by hand. Store the decanted tincture in a cool, dark place in a brown glass. Again, be sure to label this. Now that's the first paragraph, and I look at this and I'm like, I laugh. This is a hoot. Okay, now second paragraph. She's going to tell us why it works. And then I'm going to close by saying why I agree with her. The blue cohosh encourages the uterus to begin contracting and increases the force of contractions. The black cohosh helps the uterus to contract in a coordinated and effective way. The ginger focuses your energy in the pelvic area and increases the energy available to the uterus. The trillium adds its prompt and persistent influence on the uterus, speeding up the action of the cohoshes and joining with the ginger to energize the uterus. Now, if you're a scientist, you kind of look at this and go, this is really cool but kind of funny. Just all the adjectives are not very scientific. But I look at the results we got. The author says this tincture can be used to initiate labor, strengthen contractions, unstall and stimulate labor. Like if it doesn't want to go, get it going. She left. Oh no, you're right there. Placebo, boom. Okay, there we go. Deal with exhaustion during labor. Expel the placenta, help control postpartum hemorrhages. The usual dose is 10 drops under the tongue. Now, I look at this now, after several years of doing this, and I thought, she's right. At least at the level of isolated tissues, holy smokes, blue cohosh is a very strong contractor of the uterus. There's no argument with that. Black cohosh is not as strong, but instead of creating strong contractions, black cohosh creates frequency. It just kind of like helps you become a little more regular. And then ginger is not a strong contractor, but it's very steady. If you get a nice pattern that's very irregular, you put ginger on it, boom, boom, boom. It suddenly becomes very regular, like it's being stimulated. And then the trillium I haven't tested. Trillium's a little hard to get because as a flower, it's protected. Okay, so I don't know if I'm even going to chase that. I like trillium, one of our first flowers of spring, right? So they're adjectives describing what each of those herbs does matches the behavior I see on contractile patterns when it's in the smooth muscle path. So it's really like, this is incredible. And then, of course, this is a science that has been going on for years. So it makes me think we really need to start paying a little more attention to botanicals. You should like this, Amy, right? The plant world. And admittedly, when people say, I don't want to use plants, I need to use Western medicine because I don't think plants work, we have to remind them, well, where did all of Western medicine come from? Most of it came from understanding plants. So we're still working in that direction. So I need to just acknowledge lots of students who've been involved in this. And everybody contributing bits and pieces. In fact, the last 3 years, a couple students right now, Mackenzie and Brian, who are currently working on some of these projects. Lots of students. Bingo. I do want to say a couple more acknowledgments for, as a physiologist, I am not a nurse, and I've not ever worked with a midwife, but some of our own graduates are in the midwifery program. I'm thinking of Catherine Wu, who has sat down with me a couple times and helped me to connect how this could apply, and she also works in a clinic where they employ not just midwifery techniques, but also sometimes some of the types of cocktails that we've mentioned, all with an incredible amount of success. So I think it's quite exciting that the nursing program is acknowledging that and moving forward on kind of this natural medicine. But yet, and I was delighted to hear Catherine say this. I said, well, some people are going to be real worried. I'm going to have a baby and you're not an MD, all right? And you're going to give me some plants to induce labor? And I understand there's a lot of very guided application of this, depending how overdue you are. And if labor can't be induced by herbs, then we get the Western medicine back in. So it's very controlled. It's very careful. It's very, very intentional. To use a word that's overused, but intentionally, it's a very good word because they're very deliberate about when this is used and for what it's used. Because as you saw for some of these things, you wouldn't take high doses of a lot of these late-term pregnancy unless it's time for the baby to be evacuated. You could force an abortion. And so they could be misused. So a little bit of knowledge is powerful, a little bit of knowledge is also dangerous. I just think the midwife people have done a wonderful job of trying to direct this information into a applicable manner. That's it. We all have to go back to class. Next slide, any questions?
Question, Teresa, on the saponins, they're membrane permeable, but do they change membrane permeability? Do they induce calcium influx?
That's my assumption. They are very dose dependent. The more you have, the more contractile response we get. So my assumption is calcium's rushing in if they create permeation. I checked with Joy on this because she said, Joy Joan in biology, she actually uses saponins on some of her immunology activities where you want a permeating substance. So without her having tested, she thought it made sense.
Have you maybe thought about using a calcium-free buffer?
Well, we've used calcium chloride will contract uterine tissue.
Yeah, I'm just saying that if you were to flush, say, flush your apparatus briefly with a pulse of calcium-free, hit it with the saponin, if you get no effect, that would be very strong evidence that it's actually extracellular.
Yeah. I think it would work, except that we need to get spontaneous motility. Because part of the reason we do that is we have some standards. If the tissue is not showing spontaneous motility, we're not entirely confident that tissue is still viable. And in the extraction process, was it overstretched or was the animal not healthy or something like that? So it could be.
Could you do a pulse, run with calcium, normal calcium?
Oh, and then take it out and then add a calcium-free.
Do a pulse, calcium-free, and then bring it
Could be a great
back
idea.
into shape.
Yep, that'd be a great idea. Saponins brine, there we go. The other thing is we could block it, because we can block calcium chloride from creating contraction. I don't know, saponin is a huge molecule, so I don't know if it would actually still permeate but block calcium channels, so I'm not exactly sure. That I'm speculating that's what's happening, but we haven't deduced it by testing. Yeah, that's a good question, thank you. It's a good idea.
Well, I was just going to say thank you. This is so interesting. I love the joining of the old and the new. I understand there's some fetal abnormalities, especially cardiac, that can occur with black cohosh.
With black? Okay.
So I just would encourage, I'm interested in more in vivo trials with mice.
It's something to pursue. I think if I were to do something like that, I really wouldn't want to do the oral gavage route. I'd almost want to stick it in the blood vessels so I know it's there. Because that whole piece of oral gavage, unless, and this is not something I pursued, but I think somebody could. What are the metabolites of blue cohosh ingestion, absorption, that we could monitor in the urine to make sure it's actually in there, or is it just a tweak? But I didn't know black cohosh was also showing, because black cohosh and blue cohosh are not at all related taxonomically. They're two different families, I think.
A recent thing I learned from a midwife.
Okay.
So I'm just interested in really looking at the effects on the infant. And I suppose how long you're taking it prior to that immediate delivery period too. That would have, more and more ingestion.
Yeah.
If you were taking it for 2 weeks, the effect on that infant, instead of just having it work successfully one time upon delivery too.
Yeah, a lot of time. And I think the whole business, you raised such a good point, is compounded because people take black cohosh for other reasons. A lot of people, now this wouldn't be the same population, but take black cohosh as something to help handle menopause. And that's probably the biggest flavor where the placebo is. Because you can say they're feeling better or not, or I've been taking my black cohosh, life is great, I don't think I need any more, I stopped taking, I feel terrible. How do you assess if that's a placebo or not? So the thing about taking the isolated tissue out is you can say yes and no. We could take isolated hearts and put black cohosh on and see what happens, which is what we did with blue cohosh. But as a biological conscience, I kind of think I owe the mice a lot of publications or something so that they didn't die in vain. I should, yeah. A little statue of the mice. We honor them. God blesses us by using creation for intentional research to advance our own health. Thank you.
So I'm wondering about variability within these plants. So my training is primarily quantitative genetics, and so there's a lot of variation, genetic variation within any species. And so you did this barcode analysis that showed that the supplements contain the species that you're interested in, but what we don't know is what the concentration is of these active substances across. And so you talk about how the midwives use these extracts in a very deliberate way, but I would argue that they are not sure. There's no quality control within nature. So with a drug that's approved by the FDA, we know what concentration we're getting. So we know if we give somebody Pitocin what they're getting.
Exactly.
But if we give them red raspberry extract or any of these things, we don't know what we don't even, number one, we don't even know what the active agent is, which you're working on.
Yep.
Number two, we don't know what the concentration is in any given root leaf or stem. So,
and this
so
is why
I'd be
biologists take
skeptical.
chemistry. No, those are all very valid. And I can even add to that. We don't even know when we take 1 gram and crunch it up. Are we consistently crunching up at the same time? We try to customize it.
Sure.
But all of this step back to Jennifer's question says, can we just try this as it's available to the consumer?
Sure.
Because of that, now all these questions say, if we ever were going to try to have these regulated, all those questions have to be answered first. I've kind of come to the conclusion that I'm tending to trust that this stuff works as long as you follow the guidelines that midwives give. Because I think we run into all those other problems when people think more is going to happen faster or to a greater effect. But I agree with all of those are very valid statements. And we even worry about, wow, let's make a concentration, keep the same concentration the entire semester so that we don't change that factor. But we can't. A lot of times it molds. So we go, okay, we make a fresh concentration. We try to do everything the same. So we're doing the best we can to standardize, but it's a long way from the standardization that would be required for an FDA approved.
And then my other question is, what do you think is the purpose of the vodka in all of these?
It's
Calm
for the
you.
midwife.
Do you think it?
It's for the midwife.
Do you think it might help the substances dissolve better.
Yeah, and also, I think I've been told also, here at this point in pregnancy, alcohol is not going to contribute to birth damages. It won't be birth defects. So part of it is just like calm down and push, calm down and push. So, a lot of the herbals that you buy are in a tincture format. So they've been mixed up and then evaporated off and you have powders. So some of the tincture helps with solubility. Again, what we did, that is one way to go. But we don't want oils or alcohols in a smooth muscle bath. That just is not good for the bath. Also, most people are just going to take a caplet. You know, a cellulose caplet with powder. So we're trying to mimic, well, this is what they're doing from that perspective. But yeah, if you would get quite serious about being better, having more confidence that the drug is in there, I would recommend the tincture. Yeah. But yeah, the vodka is a hoot in this case, yeah.
I had just one comment on, although, you know, non-medications don't need FDA approval, there is a branding for U.S. Approval of supplements and herbals that they have met a standard for concentration. So you can see that little branding.
Okay, that's great. Great to know. Yeah, and I don't see that, at least I don't see it more because I've been trying to use the actual plant. But there is some comfort there in this attempt to address that. It's 11 o'clock, so most of us have to go back to class. Thanks for being here.